back. We are going to get started with our afternoon sessions. Hope everybody had a good lunch. So today's afternoon sessions will be pretty technically in-depth. The next two sessions have to do with drug modeling. So this is a subset model of BioGears that exists in BioGears, that is supported and validated in BioGears. So I will give a brief overview of the model, and then Matt McDaniel will kind of discuss some in-depth details of the model. Okay. And if anybody has questions, please type them in the chat, and then we will unmute you at the end of the talk, and we can have a discussion. Probably just the best way to do it. Just real quick, can everybody hear me? Somebody just wants to type in chat that they can hear me. Awesome. Thank you. Perfect. All right. Okay, so BioGears drug modeling. So what we'll do for this talk is we're going to go a little bit into the BioGears technical background, how we approach modeling the body, some of the methods and routines that determine circulation, respiration, things like that. And then we'll get into a general overview of our PK/PD methodology. How do we define substances? How do we define how they travel in the body? Pharmacokinetics, and how do we define how they affect the body? Pharmacodynamics, and then we'll just discuss a complex treatment scenario using vasopressin. So that'll be the outline. So real quick, let's just discuss a little bit about what BioGears is. And so this is kind of just a retread of what people have seen today, kind of some of our goals of the project: whole body physiology modeling, increase access to care, interfacing with medical hardware devices, encouraging open support and community involvement. Some of our strategies to meet those goals, we've also kind of heard all this, so I won't dwell too much on it, but we have physics-based lump parameter models that determine our cardiopulmonary systems. We have compartment modeling. A lot of our models are mechanistic. They are derived from base principles of some of these interactions. We have an open source library that's free to everybody, not just the library, the source code and everything we do, including our graphical user interface. We have multi-platform build testing, which is a lot of our software overhead and development costs. Our interface comes with our open standards and data model development. What does the data look like? What units does it come in? Things like that. And then supporting community development has to do with, providing software development kits, providing a forum via GitHub. We'll learn more about this later, but feel free to go in and make issues, features, enhancement bugs as much as you want in GitHub, and we will interact with you and try to get them completed where we can. So what does our cardiovascular circuit do? So we want to have compliance that mimics the pumping of the heart. So there's a lot of work actually that's been done in the background to this that we've adapted for BioGears. And then the cardiovascular circuit has pathways to our tissue compartments, which will come into play later when we're talking about drug diffusion. But these tissue compartments hold our extravascular spaces and fluids. We define our elastance changes to be time-varying double Hill relationships with some max-min constraints on them for the heart. You could imagine some extension to BioGears adjusting those max and mins, or having those be random or patient-specific. And then we set the left and right heart compliances, and that allows us to circulate fluid in the body. So you can kind of see here our pressure volume curves over time as the heart pumps. So blue is volume, red is pressure. So you can see the filling and discharging offset in the heart mechanics. Okay, so the respiratory circuit is the other key. So we want to talk about the whole cardiopulmonary system.
This is Dr.
There is a driver pressure that is variable that's coupled to our chemoreceptors. I'm not going to get into the full details of that model because it's pretty complex, and our driver has been reformatted in the past 2 releases, and our chemoreceptors have also been redeveloped in the past 2 releases. So details of those models can be found on our documentation and in some of our release notes. But what's key here is diffusion, and diffusion into the blood. So how do we get air, oxygen from the air to diffuse into our bloodstream? So we have some nice diffusivity coefficient that then scales with this pressure difference across the alveolar capillary. And so this is how we get oxygen in the blood, but it's also how we get. Thanks for the question, Dan. So we'll discuss that at the end of the talk, but I appreciate it. It's a good question. So really quickly, this is how we also get drugs into the body, things like albuterol, things like sarin, also carbon monoxide, things like that. So that is just how we get the substances into the blood. So now for our drug model, our clinical description. So first off, how do you administer a drug in BioGears? We have a lot of different routes of administration. So these include intravenous, intraarterial. You can do an infusion or a bolus. You can inject them into the musculature and have it diffuse into the blood. You can give them an oral application, and this can be OTSC, like something that they stick onto their gums and it's absorbed that way, or it can be a pill or capsule that is then digested. They can also inhale the drug through an inhaler or through the environment gas composition. You can see we have 19 supported drugs in BioGears right now. We expect this to be about 20 by the end of the summer. And we're always trying to expand the number of drugs because there are lots of use cases for them. And pretty frequently we see that as we do multi-trauma scenarios, as we work with other military groups, more and more they require different drugs for different treatments. Oftentimes these drugs are listed in clinical best practice guidelines, and so we've been expanding this library as we get more work. Okay, so how do we get drug mass from the vasculature into the tissues? So we use perfusion-limited diffusion. So for each organ, we take the flow rate Q that is traveling into that organ, and then we scale it based on a concentration difference that is then capped by our partition coefficient. So this happens for every tissue organ connection. And so as blood circulates in the body, small amounts of drug are diffusing into the tissues of every system that's present in BioGears. So from there, how do we compute these partition coefficients? We base our work off of the work done by Rogers back in 2004 and 5 and adapt it for BioGears, but this is the basic equations. These are different for strong bases, acid, neutral compounds, and actually not listed here is the Zwitter ions, so we now cover those. I didn't want to list every single partition coefficient, but you can classify a Zwitter ion and have it be computed here. So once we compute these partition coefficients, now we have a good way of getting that mass into the tissue. Now, when the mass is circulating in the blood, we also have definitions for the substance that have to do with clearance. So there are 3 different types of clearances you can define in BioGears that aren't just the tissue sequestering, which is hepatic, renal, and systemic clearance. So these come along in your substance definition schemas. So when you define a new drug, you can then, if you have an experimental value for one of these, you can input that into your model, and then we'll calculate the clearance based on that number. The FU is our fraction unbound in plasma because we want to remove unbound drug and not drug that has been partitioned onto the blood cells. So once we have a good idea of what that concentration profile looks like in the blood, we then want to kind of extrapolate to how that affects the body. And so in BioGears, what we do is we take our plasma compartment concentration, and then we look at that concentration on what we call an effects compartment. And then that effect compartment, so that's different per drug. So you can think of that as almost a delay to site of action of a drug. So we'll see a little in a next slide, how two different drugs can vary your overall dynamics. And then we have a change in effects. So there are a lot of different effects that can happen to BioGears. These are generally just due to clinical testing and treatments. Do we see a respiratory depression? Do we see effects to the renal system, things like that? And so then that's how we kind of get from our concentration profile to how it's scaled to the human body. So you can see this effect compartment coming into play with different onset, offsets of different drugs. Two opioids here being profiled for their respiration rate. One has a more rapid clearance profile. One takes a little longer to start affecting the respiration rate at the same degree. Obviously, these are two different amounts, but this is kind of just to show you that difference that effect site compartment can play in the dynamics of the drug. So putting it all together, we've got our vasculature, we've got our extracellular and intracellular space, and then we have this diffusion that provides the mass clearance into the intracellular space from the vasculature for each of our drugs. And then we have this clearance term also. And then from there, we get this nice PK profile. From that PK profile, we can update the effect site and effect concentration, and we get an effect concentration over time to what it's doing to the body. So what does this validation look like? So for fentanyl and morphine, we get a nice PK curve of the concentration profile over time. These are over hours. A lot of this work was done with UNC School of Pharmacy. It's since been extended by many different people, our work with University of Washington, our work with USISR. As we expand our drug library, we usually validate each of our drugs' effects and clearance curves with a subject matter expert who we're working with generally. Otherwise, we just try to find clinical data in order to validate this with. So Dan would like to see this figure again. Okay. And we can talk about this figure more too, Dan, at the end of the talk. Yeah, so all of the output in BioGears is done through our data request framework. So you can query most physiological metrics that you want, things like respiration rate, heart rate, but then also in BioGears through our data request language, you can access specific compartment data, so specific things like mass, concentration, pressure, things like that. So there's a lot of different ways to get data, and you can get as refined as you want. You can get the tubules, pressure, you can get the left leg mass of morphine, things like that. No problem. Okay, so now that we've kind of seen the full profile of what this looks like, let's do just a test case with vasopressin. And so this is what our XML profile looks like for vasopressin. You can see that we have pharmacokinetic parameters that you can input. So if you want to create a new drug, this is kind of where you start, is templating an XML file for the drug that you have in mind. And we want to look at vasopressin. I think it's an interesting drug to look at here because of its effects on the cardiovascular system. And and how it's used in shock therapy, hypovolemic shock, and septic shock. And so now we can look at the pharmacokinetic validation first off. So when you provide an infusion of vasopressin, so for this one, what we were able to do was find a clinical paper that gave us a nice time concentration profile for vasopressin. And these were just male volunteers, so this is oftentimes an issue that we run into, is that clinical trials don't profile for the entire population always. But this is sometimes okay for BioGears. You know, we do our best to validate what we can. So for this scenario, we infuse 0.174 micrograms per minute over 60 minutes, and then we, in their observations, they observed how it was cleared over the next 60 minutes after that infusion. So that's our PK curve. So now what we wanted to do was look at its role in hemorrhagic shock therapy. So we were able to find some rationale for this. So there is ideas that vasoconstriction can improve organ perfusion, especially when there's a really low amount of fluid. There are some risks involved in this, and there has been some successes shown with animal studies. And so we were able to find, with the help of University of Washington, a couple studies that profiled the use of vasopressin versus a control of just saline in patients who had had a very severe hemorrhage. So the idea of this whole thing is like, okay, I see this clinical study, I can now profile that study in BioGears, and then I can simulate, I can do two things. I can either decide to simulate a bunch of patients over the same study, kind of creating an in silico clinical trial, or what I can do is start varying some of the treatments. So do we have as much vasopressin for one treatment versus the other? Things like this you can rapidly profile in BioGears because it's an in silico model, so you don't have to go through all the framework of setting up patients and other things that you would in a clinical trial. So it's a nice first pass to do some of these investigations. So this is what the timeline looked like for the study in 2011 provided by Cone. We're going to have one group that just is given saline and another group that is given saline with vasopressin, and they're administered at the same volumetric rate. So from there, we have our initial hemorrhage, and you can see from these graphs, these were some of the outputs from BioGears that we got that we then validated to that treatment study. And a lot of these profiles actually were pretty consistent with the study. We don't have all the details here on this slide, but we actually have a publication where we talk about this more that was published at the beginning of 2019. And I believe it's called like BioGears Tutorial in Pharmacology or something like that. So look that up if you want a bit more details on this. But basically, you can see that we have a vasopressin group and a control group. And each of these groups were hemorrhaged extensively, so very severe hemorrhage. And then one of them was given vasopressin, the other was not, just given saline. And you can really see the responses in the mean arterial pressure rebounding. You can see that it drops due to the extensive hemorrhaging and then rebounds after that. The heart rate, you can see it starts recovering in the vasopressin group due to the MAP changes. And then the filtration rate, which is a really nice indicator of end organ perfusion, really recovers well for the patients who are given vasopressin. So pre-treatment, BioGears captures the feedback responses to massive blood loss. So these are the drops in MAP, the things that are happening due to that massive hemorrhage that the inpatients had. You can see our baroreceptor-mediated increase in heart rate, our decrease in filtration and urine output. And then our treatment, you need less fluid to achieve the MAP increases that you would want in your patients. We have tachycardic reversal, increases in urine output, so we have a really nice idea of how end organ perfusion happens. Some of the improvements that we'd like to see in BioGears and that we've actually added since that study in In 2019, we've added some minor adjustments to our baroreceptor. We don't have the full decompensation that you might see when a patient like totally collapses at the end of a large hemorrhage like that. And we do have site of action pharmacodynamics, but I say this specifically to vasopressin because the vasoconstriction may not be so systemic. There's some evidence that it may only be certain organ arteries that are being affected, certain smooth muscle cells, and not every smooth muscle cell in the body. So right now, our MAP increase is a function of like a systemic vessel constriction, where this may not be the case, and this is absolutely something that we could integrate into BioGears and would be a really interesting extension. So just an overview of the physiologically based pharmacokinetic model. We have a lot of different effects. I didn't list them all here, but things that you can do with drugs. I think we have about 10 or 15 different pharmacological effects that can affect the patient. You can have delayed onset in terms of site of action. You can give them drugs via multiple routes of administration. It's fairly easy to extend the model to include new drugs. The validation is usually maybe the harder part. And then the real benefit of all of this is that you can construct these complex clinical studies and you can run them over and over again. There's no overhead. Once you have that constructed, once you're getting data on the other end, out on the other end of BioGears, you can really run through these studies pretty rapidly because of the speed of the engine. So yeah, and then we will have a GUI that's coming out in the next few months over the summer, and that should remove a lot of the burden for templating some of this stuff. So do you want to give them just a drug, see how it affects somebody, maybe give them a few interactions like a hemorrhage or a lung collapse, and then give them ketamine or something like that? You can kind of explore these physiological scenarios with that user interface without needing to build BioGears or download the libraries and understand the executable. So thank you. And if anybody has any questions, I think Dan had one. So I will. Okay, let me unmute Dan first, and then we'll go to Rachel. All right, Dan, you are unmuted.
I just wrote it up there, but how does BioGears address multiple medications at the same time? So if I gave vasopressin, but I had already given atropine and maybe some epinephrine, how does it coordinate those different effects?
Yeah, so this is a great question, and one that we've done a little bit of development on. So we do have a drug-drug interaction model right now, but it doesn't extend necessarily to every drug. But in the example you gave, though, our pharmacodynamic curve is a hill-type model. So you will saturate on sort of the upper end of what you call like an overdose for those given effects. So if you were loading epinephrine, vasopressin all onto the patient at the same time, you'll reach that saturation point for those blood pressure effects. So that's one way. We have a more complex model, but we've only extended that to things like naloxone and opioids in terms of reversals. So having this antagonist that are trying to bind to the same site as your opioid, we've modeled that. And then for sarin and pralidoxime, we've done a similar thing where there's this co-competitor binding. But yeah, so we would like to extend that a little bit to some other models, but that's a great question.
Thanks.
And then for your question about the validated for physiology, if you go to our website, you can see all of our validation metrics, and generally we try to validate pressures, flows, volumes for most compartments in the body and provide references for all those metrics. We're always updating those tables, so just go to biogearsengine.com and you can kind of see the extent of some of our validation.
but in follow-up, the struggle is that you really can't, because it's so hard to do these studies. I was trying to do epinephrine, for example, and finding a clinical study that actually gave a dose of epinephrine and followed those physiologic parameters. Uh, the only studies I could find were in dogs, and they were 30 to 40 years old. No one's doing that science anymore. And you probably have access to different capabilities than perhaps I do. But are you running into that same problem? Over.
Yeah, absolutely, Dan. Like this is a great question, and in terms of validation data, this is a difficult question. Yeah, for drugs, like we're not just going to expose a bunch of patients to sarin and see what their biomarkers are, right? Like that's just not something that's going to be approved ethically. And so oftentimes we are searching a little bit, and we will try to grasp onto things like animal studies and then do some statistics to try to get a reasonable idea of what a human patient would look like. Other times we find like really bizarre reports in publications where, like for example, we validated, I think, our morphine or fentanyl, where a patient had a broken pump on a self-administered opioid, and then they were just giving themselves a lot of that opioid, and it wasn't rate limited, and they happened to be in the ICU and happened to be having their metrics collected. So yeah, I mean, drug modeling is difficult. There's other ways you can kind of do it, statistical machine learning ways that you can integrate in order to find like similarities between drug profiles and chemical compositions. You can look at how some of the drug is sequestered in different organs. But yeah, I mean,
I'm not suggesting that you guys aren't doing a fantastic job, because you're really trying to validate something that's very hard to validate. But even in that example that you said, you've got an N of 1. And in that, you're validating against a morphine curve on a person who may have been a very sick person with a cardiac ejection fraction that is not normal, or who knows what his underlying renal function was. So it's really a challenging thing that you guys are trying to do. So I'm not downplaying, but the problem that I see is people keep saying, well, what validated physiology engine are you using? And really the validation, even the best validation that you can do, or Pulse or Pure Med Sim or anyone, is pretty challenging and not 100%,
Oh, yeah, definitely. No, that's it's a really great statement. I think, there's a whole project out there, right, that is literally just validation, no new models, and do that for 2 years. And, and then it's interesting when we were developing our burn model, especially when you come into trying to validate multi-trauma scenarios and then drugs being administered during a multi-trauma scenario, there is literally zero data. And so we were working with USISR, and I mean, the only way we could do it was just to iterate with their subject matter experts in terms of, okay, you've got a patient with a pretty large hemorrhage and a large TBSA burn, and you get them 2 hours in and you give them ketamine. What happens, right, to their physiology? And there's not going to be any paper that you can find that's going to tell you. But there's a real nice intuitive sense that somebody who's been on the front lines in an ICU tent has seen, and they kind of have an intuition, right, of how they think a patient might behave. So it's like kind of extrapolating some of that data from them is how we try to do it.
You are using some expert.
Fuck up, Dan.
I'm sorry. This is the last thing I'll say. You are using some expert validation too, which isn't, some subject matter expertise validation, which is the best you can do. All right. Thank you. Sorry for taking all your time.
No problem. Rachel, you want to go?
Yeah, I just had a general question about the future of BioGears and if you ever think it will be able to model cardiology in any way.
Do you mean in like a classroom sense?
No, in a medical simulation. I mean, we use BioGears to model trauma simulations, but it restricts us because we can do no cardiology simulation.
Okay, so like
And I know it's incredibly complex, and that's why I'm wondering if that's just not even going to be a realm BioGears is going to venture into, or if it's even mathematically possible.
Yeah, I think mathematically it's possible. We could definitely get into changing the heart dynamics, changing ECG rhythms, adding electrocardiology to some of the signals of our pulsing and our myocardial cells. We could do all that, absolutely. We don't support that currently. I think it's a great question, and really would be a nice area to move into. We just haven't been able to find sort of the collaborators to get there.
Okay, thank you so much.
Thank you. And if anybody has any questions, otherwise I will hand off the presenting to Matt, and Matt can go over some of our newest drug models that we've just integrated into BioGears. So